Welcome back to Student Research Week on Across the Fence. Here in the Karametsov lab at UVM, researchers study the role of genes and the environment in autoimmune and infectious diseases. And the insights of one undergrad could underpin future therapies for combating destructive bowel inflammation. Let's go learn more. Hello, Vermont. In labs all across UVM, undergrads and graduate students work side by side on research teams, learning how to do research while simultaneously pursuing their own novel questions, pushing the parameters of a given lab's field of inquiry while furthering the process of discovery. My guests today, one undergraduate, one graduate student, joined forces in the Karametsov lab 3 years ago. One of them will be presenting a poster on their work this week at the Student Research Conference. Say hello to Julia Young, an undergraduate majoring in molecular genetics at UVM, and to Tyler Hogan, a third-year PhD student in the Karametsov lab who's been serving as her in-lab mentor. For the record, Tyler is a lifelong Vermonter who also did his undergraduate at UVM and won me over by talking about watching Across the Fence growing up. I liked him immediately, so. All right, well, Tyler and Julia, it is wonderful to have you here. I also just want a quick note. It's the Karametsov lab. I said it incorrectly in the introduction. Apologies to Dr. Karametsov. But Julia, tell us about your work. What have you been studying?
So I am studying how natural genetic variation in a gene called NOS2 affects the function of a protein called iNOS. And iNOS, its entire job is to create this gas called nitric oxide in immune cells and in the body. And that nitric oxide then goes on to damage and kill microbes that are infecting you. And so that might sound like a really good thing, like you want as much nitric oxide as possible. Right. However, it's a little bit more nuanced than that. The nitric oxide that iNOS produces can actually go back and damage host tissue and host cells. And so this has even been linked to predisposition to disorders like inflammatory bowel disease. And so it's really important to regulate this iNOS activity and nitric oxide production. And it's even been referred to as a balancing act, where you want enough to control an infection, but not so much that you end up hurting yourself in the process. And so I'm studying how this balancing act is regulated at the gene level.
That's fine. It's like a Goldilocks type situation.
It really is.
You want to have exactly the right amount.
Exactly.
Yeah. So Tyler, how did you begin working with Julia?
Yeah, so I started working with Julia, was it about 2 years ago,
Yeah,
3 years ago,
yeah.
when I was a first year graduate student, or actually, was it before I was a graduate student and working as a tech in the lab?
It all blurs
Time
together
blurs
at some
together,
point,
yeah.
And
but
because you've been in the lab for quite a while, because you did
Yeah.
undergraduate, and then you were a lab tech, and then
Yes.
you've been a PhD student.
now.
Yes.
So yeah, time blurs together really, but it's a great experience.
And Dr. Karametsov put the two of you together.
He did, and Julia actually worked with a graduate student that I worked with in the lab, Brishji, who's now at Harvard doing research in neurological disorders and the immune system. But Dimitri put us together because we were interested in pursuing this genetic variation in iNOS, and previous work that I had done was to characterize what are called variants of unknown significance in a tumor suppressor protein called STK11. So I've had experience in characterizing different variations in proteins. So that is kind of why.
It's something interesting about the lab environment, because oftentimes there's synergies between projects, but they aren't necessarily going after the same question.
Yes.
Right, so there's like in this lab, for example, I think you do a lot of work with multiple sclerosis?
Yes.
Right, okay, yeah, so, but finding the synergy, which doesn't necessarily have to do with inflammatory bowel disease, but there are all these different overlaps in the question. Pretty cool. So Julia, how did you end up in Dr. Karametsov's lab?
Yeah, that was a pretty classic story, I think, for a lot of college students. I was interested in getting into research, and so I was looking at different lab pages, and I came across Dr. Karametsov's lab page. And some of my favorite things in biology in high school and early in college were genetics and immunology. I just love the fact that genetics, I feel like it's really the cornerstone of cell biology. Genetics is what instructs everything in the cell to do what it's doing. And then I was so fascinated by the immune system and how this vast array of cells all have this really intricate dance and process that they all work together on, and we don't even know what's happening. And it's happening in our bodies constantly. And so
you just reached out?
Exactly. Finding out that he was an expert in genetics and immunology was perfect. And so I sent him an email and I explained my interests and what my goals were. And we had a one-on-one meeting and discussed it a little bit more. And he agreed that our interests were really aligned with one another and that my goals would be a good fit in the lab, and so he offered me a position.
Well, indeed, it is a good fit in the lab, and we're going to take a look at it now because Julia and Tyler let me follow them around the lab one afternoon to see what a day in the life is like. Let's take a look.
Day in the lab is really different every time. It really depends on what needs to get done, what the next steps of the experiment are, so it's really exciting. Time flies. I sometimes am not aware of how long I've been in the lab, which is kind of crazy. I think my favorite activity is cell culturing. I love taking care of the cells. I think it's really cool to see living things in the microscope and to take care of them. And it's probably my favorite, just the idea of taking care of living cells. I think that's really fun. And so I really love splitting cells and doing any cell work at all. These are human kidney cells, and that's what I kind of use to do all of my experiments. The platform for running all of my experiments and doing all my tests and stuff. So to take care of them, I want to start by suctioning off all of the old cells and all the old media. Trypsin loosens up the cells that are stuck to the bottom of the flask right here. Lets me lift them up and get them back into solution. So I'm going to pipette a couple milliliters of that into the flask. And now this part, we put the cells back into the incubator for about 5 minutes and let them just kind of sit in the trypsin and let all the cells lift up. So these are the cells that are now trypsinized, so they've been treated with the trypsin enzyme and they've lifted off the bottom of the flask and now they're floating around in the liquid. So you see all those kind of whitish clumps in there, those are actually human cells in culture. I think, especially being an undergrad and being surrounded by so many really smart and really accomplished people, it is really motivating to be in this environment and to see how many people are working so hard and doing such amazing work, and it really makes me want to work harder as well.
Okay, time flies in the lab. How much time do you spend in the lab each week?
It really depends week to week. There'll be, over the summer, I was a full-time researcher. I had a funding award, so I got to stay in the lab full-time, which was fantastic. And I would definitely spend up to, I think, maybe 30 hours a week in the lab. Yeah. During the school year, it's a little bit tougher. I try to spend at least 9 or 10 hours if I'm working on experiments, try to fit it in. Sometimes it'll be a little bit more than that if I have more meetings or longer experiments. Sometimes a little less if I just have to do some data analysis.
It's a lot on top of all of your schoolwork, I'm imagining.
It can be, yeah.
Okay. Well, Tyler, okay, so you're pursuing your own research at the same time as you're working with Julia. So are your research interests complementary?
They are. My kind of broad interests are how cells communicate with each other, and nitric oxide, in addition to killing bacterial cells, is also a signaling molecule, so it also is involved in regulating other cells in the body.
Ah, okay, and so what do you get out of the process of mentoring a young researcher?
Yeah, so mentoring is something that I've been involved in ever since I became a lab technician, and I really appreciate working with undergraduate students. Not only does it help me keep on top of the science so I can explain what's going on and educate them, but it also, it's a very positive experience to see young scientists grow up and gain the skills and thought processes that
And they really do say that you understand a topic differently when you've had to teach it.
Yes, that is 100%
So
true.
you're going to be presenting at the Student Research Conference. Tell us a little bit about your poster.
Yeah, so I will be presenting a poster on the thesis project that I've been working on for the past couple of years. And my project, like I said before, is all about NOS2 and iNOS, and I'm specifically looking at how these two different lab mouse strains vary genetically and how that genetic variation in the lab mice leads to differential activity in their iNOS proteins. And I think it's kind of important to note that looking at lab mice, it might sound like, why aren't we looking at humans? But because so much research is done at UVM and across the world in labs using mice as a model organism, it is really beneficial to understand how everything works in those model organisms, so we understand the science better and can apply it to humans. And so I think probably the coolest finding that I'm going to be presenting at the Student Research Conference is we were able to identify one letter change in the NOS2 gene that corresponds to one amino acid change in the iNOS protein. That actually leads to about a 35% reduction in iNOS activity, which is pretty stunning. Yeah.
And pretty exciting that this was your discovery.
It was.
That's fantastic.
Yeah.
Tyler, when did you know you wanted to do research?
It wasn't until I was really a lab technician that I was committed to doing research. I entered undergraduate thinking I wanted to go to medical school. Growing up in rural Vermont, I didn't have that much access to science and know really what a career in science looked like. I really only knew medicine as a option. But as like Julia, I was in the honors college and did an undergraduate thesis, so I started doing research in undergrad and then started as a lab tech once I graduated, and then really just fell in love with researching.
I think that's often how it happens. And then Julia, you said, and I'm going to kind of quote you back to yourself, that you knew you were hooked when you realized that you were the first person to know a thing.
Exactly, yeah. I had a very similar story to Tyler, tale as old as time. I really thought that loving biology meant medical school.
Sure.
And so actually, Bristy was the first person to, that old mentor that Tyler mentioned, who said to me.
And this is your plan after graduation, right? You're
Yes,
you're
I'll
staying
be
with
doing
research.
But yeah, I think I really fell in love with the whole, you know, it takes a while and it takes a long time to get a result, but then when you finally do get that result, you're the only person in the world to know that one thing. And it's a great feeling until you share it.
That is really incredible. Well, Tyler and Julia, thank you so much for joining me today. Appreciate learning more about it. Good luck at the poster conference. It's going to be great. And as always, thank you, Vermont, for learning and exploring with me today. And thanks to the team that makes Across the Fence possible. I'm Amy Finley. This is Across the Fence. Come back and let's learn and explore again tomorrow.